Okay, this is again an extended concept on ELISA. All of you know what is an ELISA, enzyme linked immunosorbent assay, an analysis in the microbiology laboratory which is based on antigen antibody reaction and specific character. So we have two types of ELISA discussed in the lesson as the picture shows. A direct ELISA. Okay, so you can see here, first we coat the antigen on the surface, adsorption. Then we add an antibody with an enzyme and we give the substrate to an enzyme, and whichever performs catalysis will give the color, is the point, isn't it? So let it be one antibody, let it be two antibodies. In case of the first picture, also the antigen is in contact with antibody. In sandwich ELISA also, the antigen is in contact with two antibodies. Okay, so because the antigen is directly in contact with antibody, this is called as direct ELISA. Direct ELISA, because the antigen is in direct contact with antibody. But you have another type of ELISA which says indirect ELISA. Indirect ELISA means the picture shows indirect. Here also we use two antibodies. Okay, here also we have an antigen coated onto the surface, and this is one antibody and this is another antibody. So we have two antibodies with antigen. So if we had two antibodies with antigen, we studied in sandwich ELISA, where both the antibodies are in direct contact with the antigen. But here if you see, first you coat antigen in the first step. Second, I add antibody, which is the first antibody. Then we add the second antibody, which is given in yellow color. The second antibody is enzyme linked. The second antibody is enzyme linked. So the second antibody is not in direct contact with the antigen, in contrast to sandwich ELISA. It is indirectly in contact with antigen, but this is specific to the antigen. So the two antibodies are chosen in such a way that they can be specific to the antigen, but still they can combine one after the other with the antigen. Now we give the substrate and the color change gives me a positive test. Since one of the antibodies used here is not in direct contact with the antigen, instead it is contact with the first antibody, this is called as indirect ELISA. Now these are the transformations in ELISA. Sandwich ELISA is more specific. Indirect ELISA is also more specific. Direct ELISA is also specific. So the entire ELISA test is transformed to find these, but all of them are performed in the same way.